All right. Mr. Ward, now is not the time for that.
There's no other
There
one.
must be the Sharpenizer XP 2400 Stealth Autumn.
Expensive edition.
Excuse me. Go search for it. We have to have a lockdown if we lost that because somebody may have ran away with it.
I
And
took it. I took it and put it back on the counter.
the thing, there would have been a school-wide investigation if that had gone missing.
I emptied it. Who had it? Oh, I was emptying it.
Jonathan was emptying the pencil shaving receptacle.
Were we supposed to write down fries?
No. All right, let's go get moving here. How are dissecting and compound microscopes different? Nicole?
The magnification is different.
In what way?
The compound used 40 times 400 times, and then the dissecting is 10 times.
Yeah, the compound microscope gives a much greater level of magnification. That's good. What else? Courtney?
The dissecting gives depth.
Yeah, the dissecting microscope gives a sense of depth to your image. The compound microscope is a very flat, two-dimensional view. Morgan? Two eyepieces, I think you mean. The dissecting microscope has two eyepieces. Compound microscope has a single eyepiece.
The dissecting has one lens, and the compound has 3.
Objective lenses? Yeah. Actually, though, our dissecting microscopes, some of them have a objective lens in which you can rotate it to give different levels of magnification. So it's a little bit different, but yeah. All right. Let's move on here. What we're going to be doing. Question?
Do we have to write down horizon?
No. We use that spot today. So, couple things. We're in our microscope section. We'll be finishing it next week. Couple things. You'll have a lab due on Friday. You'll have a quiz on Wednesday of next week. I will not be here Monday or Tuesday. I'm going to be at a conference. So you'll be working on some microscope stuff with the substitute. Okay? Topics for that quiz are online. You can look them up there. You'll be doing a review sheet on Tuesday, but you can see that information online. All right. So today we are going to start by just finishing up all of our microscope notes, talk a little bit about measuring with the microscope, and then we'll start to get some practice using the microscope. So that's where we are going here today. So take out your note packet from your well-organized, numerically ordered, soon-to-be graded binder.
Still upside down.
Anthony, is that microscopes? Is the first page in your binder?
Yeah, I'll fix it. Can I go to the bathroom?
All right. All right, so just to give us sort of a roadmap so far in this unit, we've talked about the various types of microscopes. We looked at some sample images. Then we talked about the parts of the compound microscope, and you're going to have to know those parts and what those various parts do, what they're used for. Today we're going to finish up by talking a little bit about field of view, some general information about microscopes, and then we'll talk a little bit about how we measure things using a microscope. All right, so first off, just some important information. It's called a compound microscope. Why? We talked about this the other day. Joey?
Many parts to it.
Yeah, and not so much the many parts, but Kristen?
More than one.
Yeah, more than one what?
Lens.
Lens that magnifies the image. Two lenses, actually. When we use our compound microscopes, the image is being magnified by two different lenses. As light goes through the microscope, first it's magnified by this objective lens that you have selected. And then as the light travels up through the body tube, it gets magnified a second time by the eyepiece lens. So there are two different lenses that magnify the image you're looking at. The other thing to notice is that because of the arrangement of lenses and mirrors in the microscope, it actually gives you an image that is flipped upside down and reversed right to left as well. Now, one thing that you're going to have to do is to figure out the total amount of magnification as you are looking at a specimen under the microscope. So the image gets magnified twice, once in the objective, once in the eyepiece. So anyone know how do you figure out the total amount of magnification there would be? Bianca?
You add the magnification from the eyepiece and the magnification from the objective.
Not quite, because if you think the objective lens makes your image certain times bigger. Then when that goes through the eyepiece lens, that then makes it certain number of times bigger. So what would we do, Jonathan?
You would multiply.
Yeah, you multiply the power. So the total amount of magnification as you're using the microscope is the power of the eyepiece times the power of the objective lens. So for example, if we were using a 10x eyepiece and a 30x objective lens, the total magnification would be what? 300, yes. Mr. Richards would be proud. 300, 10x eyepiece times 30x objective. That means that what you are seeing as you look through the microscope is enlarged 300 times larger than real life, than that object actually is. Another important thing to keep in mind as we are working with microscopes is how to properly carry a microscope. Okay? You always want to use two hands, holding on the arm and the other underneath the base so that you can transport your microscope carefully. They are fairly expensive. They also are fragile in terms of if you knock them around, they get out of adjustment, and it makes viewing things much more difficult. So you want to be fairly gentle with the microscopes.
Wait, what was the last
One hand on the arm, one on the base. Now, whenever we do work with a microscope, it seems like almost every group has their hand up constantly saying the same thing. Saying, I don't see anything. I can't see the object. If you follow the proper procedure, it's really not difficult, but you have to really follow the steps in order to be able to do that. So that's what we'll talk about. So first off, we always start on low power. Whenever you're going to begin viewing something, you will always start on low power. That's the red objective lens, the shortest one, the 4x objective. Then you place your slide. You always are viewing, in the compound microscope, we're always using a slide to view the object. So when you place your slide, you clip it in using the clip there. And you need to make sure it's centered. You need to make sure the part of the slide you want to see is right over the light. Okay? The other thing is, when you switch from one objective lens to the other, as you rotate the nose piece, there's notches. It has to be properly clicked in. You'll feel it when you switch from one power to the other. If you're not quite clicked in, or if you're halfway between, you don't see anything at all. The same is true of the diaphragm. When you switch settings on the diaphragm, the light gets brighter or not as bright. But anyway, if you're between two settings, then no light is going to get through. So you got to be clicked in on both the diaphragm and the objective lens. And once you have it, then you focus the image using the adjustment knobs. Yours is phrased slightly differently. You're going to use the rough the coarse adjustment to focus. And again, on our microscopes, the coarse adjustment is this larger part of the knob that focuses the image. So once you have your slide on, you're under low power, it's centered, you look through the microscope and then you adjust. Okay, you adjust the focus. At some point, you're just looking through, at some point, the image will come into view. And you make sure that it's very clear, that it's focused, and then you can draw if you need to, or look at your specimen. Under low power, you can see a lot of the slides, so you can scan around to see what parts you want to look at in more depth. So then, if you have it under good focus and it's centered, then we can switch to a higher power. It's important that you have the part you want to zoom in on centered in the field of view. You want to make sure it's in focus, and then you could switch to the medium power objective. Okay, the medium power objective is yellow. It's 10x. And again, once you make the switch, if it was in focus under low power, it should be very close under medium power. You may have to make some small adjustments with the coarse adjustment knob, but it should be really close. Okay, and you could, again, look at your slide. If you want to look even closer, then you would center the part you want to look closer at. You make sure it's in focus, and then you could switch to, finally, high power. Now, high power objective, the longest objective lens, it's blue, it's 40x. It gets very, very close to the slide. In fact, the thicker slides, it almost or sometimes does bump into it. When we're on high power, we only are going to focus using the fine adjustment part of the knob. We don't need to move it very much at all to focus under high power. So we only use the smaller part of the knob, the fine adjustment, to get it in view. Okay? Now, one thing to make note of. So first, we're looking at something under low power. Again, first we center it, focus it, then we could switch to medium power, focus it, center it, and then you could switch to high power. And last thing, because again, just like it flips the object left to right and upside down, when you are moving something in the microscope, it appears to move in the opposite direction. If I'm using this microscope and I move the slide to the right, when I'm looking through the microscope, it appears to move to the left. When I'm looking and trying to move it forward, under the microscope as I'm viewing it, it seems to move backwards. So things are reversed in that situation. All right, so in our microscopes, the powers of all those microscopes back, they're all the same. Oh. And so when we are on low power, this is our field of vision. A field of vision is sort of the area you see. So under low power, we would see all of this. This is a paramecium. And so we start off viewing in low power so we can see all of these slides. We could scan around, find a part that's interesting, and then look closer. So this gives us a wide field of view. Now, as I switch to medium power, how much of this am I going to see? No, Nicole? Yeah, about half. I'm going to see less, though. That's the point. So as we move to a higher power, we see less of the object, right? Because we're zooming in, we don't quite see as much. When I switch to high power, again, I'm going to see even less of the object, okay? Because I'm zooming in, zooming in, zooming in. So we say our field of view gets smaller as we increase our magnification. So in our microscopes, when we have the low power objective lens set, what is our total magnification? 40. Under medium power, it's 100. And under high power, it's 400x. So what that means is when we're under high power, what we're seeing is 400 times larger than its actual size. And again, field of vision is the area you're seeing. As you look through the microscope, it's the area that you see. So as we increase the level of magnification, what happens to our field of vision? Nicholas? It gets smaller. It decreases. And if we decrease our magnification, our field of vision increases. There's an inverse relationship between these two. All right, so just some more examples. Again, low power, what's our eyepiece power? When we are using low power, the eyepiece is what? Eli? No, the eyepiece power is 10. What's the objective power under low? Emma? 4. For total magnification of? 40x. So low power gives us 40x magnification. It gives us a large field of view. We can see a pretty wide area when we're on low power. Also, as an example, I know it's Halloween, so I thought I'd give you some scary, gross monster pictures. Sorry if I gave you a fright there. So, if we are going to switch to medium power to observe this bizarre creature, then what would our eyepiece power be? Chloe?
10x.
Again, it's still 10. What would the objective power be?
10.
Also 10 for total magnification of?
100x.
100x, giving us a medium field of view. I understand if you need to turn around, you can't see this. All right, sorry to do that to you, but it is Halloween. And finally, if we're going to zoom in on this disgusting creature under high power, what would our eyepiece power be? Jonathan? 10x. And objective power?
40x.
And the total magnification?
400.
Yes. Giving us a very small field of view. We could see much less of this.
You! You get it away.
All right. Any questions about using these microscopes? All right. I'll take this off the screen so you guys don't have to be subjected to this.